Okay, good morning. Hello. Before we start today with talking about event-related potentials, are there any questions that are left over from Wednesday, both with regards to our course and maybe also with respect to the stuff that you then saw in the afternoon? Anything that you were confused by or that you want to remark on? Or was it all just too overwhelming and too much?
Regarding electricity, can you find everything you, or is most of what you mentioned in the lecture in these books that you mentioned before?
Yeah, I mean, with the electricity, the stuff that you want to really learn by heart is on the slides, and everything else I mean, Wikipedia is also a place to start to learn about electrical fields or electric potentials and so on. And then, of course, there are many physics books that discuss these concepts all a little bit different. But in the end, the stuff that you should know is on the slides. So that's the bottom line.
The formula and stuff and things we need to understand. I have zero basis, so I understood nothing.
No, then you need to work.
That's all.
It might also help with respect to formulas and stuff to start working on the mathematical preliminaries for statistical methods also, because this covers also a lot of the basics in terms of mathematical language and writing. So I can only recommend that. But of course we will talk more about statistical methods in 2 weeks. Then in terms of Wednesday's stuff, maybe 2 remarks. First thing, Rainer Göbel gave this talk and he sells the software Brain Voyager. That's a little weird. So as a scientist, you don't sell your software that you write to other people. You do what we call open science. You share your code because you already get paid by everyone to do this work. So I just don't want to now leave you under the impression that everybody in science would do something and then sell it to other scientists. That's weird. That's fairly old school. The other thing, maybe with regards to all these young people that do some modeling and then don't talk about the models, that's also not necessarily always the case. It's just 2 things that when I listen to these talks, that sometimes makes me a little bit crazy, so that everybody says, and then we use this awesome model, which I don't explain right now, but we used it, and these are the results. So this is also something that needs to change in cognitive neuroscience, because there's just not much progress if everybody's always only focusing on awesome results. Just 2 things that came up in my mind when I listened to these talks. But otherwise, I hope you had a little bit of fun, you learned a little bit, and you saw everyone involved. Any other questions, comments on Wednesday? Because otherwise I thought it was nice. It was kind of because it was a public event later, I think it's maybe I don't know where you learn more, whether it's such a public event style kind of thing, or you learn more if you really hear the normal talks, although the normal talks in science often are also like these talks. Anyway, good. And with this heavy mood, let's go into event-related potentials. So we ended up on the neural origins of the EEG last time, and I said that basically this slide here contains most of the information. So is anyone up to explaining what this slide is about and what we've learned last time? Just go for it, and nothing bad will happen, because this is not the exam. In the exam also nothing bad will happen, but if you don't have anything to say in the exam, then you will not pass the exam. So, any suggestions of what this slide? You actually see that? I mean, we can put the shades down, but then it gets really dark. I don't know.
I think it simply shows intracellular and extracellular activity of the neuron during the synaptic transmission.
Yeah. So basically, what this is supposed to indicate is that there is some synaptic activity, and then in this case inducing excitatory postsynaptic potential, which leads to positivity inside the cell, which then is accompanied by negativity outside the cell, and everything happening simultaneously here at the apical dendrite. Because there's a lot of negativity here, current needs to flow, and there's a simultaneous positivity at the cell body. So this basically introduces how at the level of a single cell you get extracellular currents and the accompanying potentials. And then what was that about? So what?
Just that there are many pyramidal cells who are aligned in the same way, and therefore the dipoles of each individual pyramidal cell add up to one larger dipole. That's directed towards the inside of the cortex.
Yeah, in this case, definitely. So the point really being, like you said, that there are many and that they are simultaneously active in a circumscribed region. And then this dipolar configuration basically maps and due to the currents that are conducted throughout the volume can be observed on the skull. This is again, as we will also see today, this is really the model for early sensory activity. So when you have something coming in and then being relayed, let's say in the primary sensory area, the idea is that these kind of things then also happen if things get more complex at later stages of processing, but really the experimental evidence and that the data really shows these kind of stuff we will see holds for primary sensory areas. Okay, so that's this simple model of the neural origins of the EEG, and like I said, it's likely, and there's also we had one slide that things are likely more complex than this, but there's not that much research right now looking into these from the cellular to the mesoscopic, so from the microscopic to the mesoscopic to the microscopic. There's not that much research because cognitive neuroscientists mainly focusing on recording the EEG and not other activities. Good. So that was basically the biological origin. This slide we've also already seen, and I brought this up again because we will today see many examples for event-related potentials. So anyone feeling like summarizing what we see here and what certain points are? Please, come on.
In the bottom we see the activity or the average activity of one electrode over time.
But it's not. What do you mean by average here? Because
average over trials.
This is actually not. I mean, could be, but this is actually not the average yet, right? So,
okay. But it shows that we always cut out the critical time around stimulus onset.
Exactly. Yeah. So so as you see here, this is 9 seconds, and during these 9 seconds, 2 X's and 2 O's, and again 2 X's are presented. And as you rightly said, this is one electrode. So these are. So these electrodes commonly now recorded with 64 electrodes. So a number more than these here, which might be 16. No, that's more than 16, right? 32, it looks a little bit weird. They have names, and they have these, they are basically anatomically named, so frontal electrodes, temporal electrodes, central electrodes, parietal electrodes, and occipital electrodes. We will go about this naming conventions at a later point. But yeah, as you said, there's for one electrode, there's one time series of potentials, we call it from the scalp, and this peristimulus time window or critical time windows, they are cut out from the data, fairly straightforward process. And then what you said earlier in terms of the averaging, that's then symbolized here. So here are these, from a single electrode, these epochs or peristimulus time series or time windows, however you want to call it, and then the resulting average. Yeah, so that's the logic that will be behind all of the event-related potentials that we see today. Yeah? So you always should think these event-related potentials are the average responses to a number of stimuli, usually in the order of 100, 300 or so, depends a little bit on the modality and so which sense you're actually looking at. And they are, if you see this kind of thing, they are usually referring to a single electrode. Sometimes you also see these kind of plots where the trace has been averaged over multiple electrodes, and this is the average over multiple electrodes. But the important thing to keep in mind is that the activities on different electrodes look different, and we will also see that. Because I found that a little bit confusing in the beginning when I learned about EEG, that you often see these kind of traces, but you know that they have many electrodes on the head, so what happened with all of the data? But that's the event-related technique, event-related potential approach. Good, so then we are now interested in learning a little bit about the experimental effects that you can observe using the EEG technique. And these experimental effects or event-related potentials, they are also the basis of many if you want, more advanced studies that might happen today, where people are interested, for example, in explaining trial-by-trial variability. So you might remember Mario's Philostides talk at the beginning. So he talked a little bit about trial-by-trial variability. We have also done some work on that. But the thing is, I usually say that if you don't find something in the average, you can be lucky and find something on the single trial level in terms of differences between conditions, but more often than not, you are not lucky. So having something if you have something in the average, then that means that must come from somewhere, and then you will have differences also at the single trial level. If you don't have anything on the average, you must be very, very lucky to find something robust in terms of condition differences over single trials that maybe at one trial are lower than the other. What I want to say is that even for the more advanced, fancy stuff that is being done, the bottom line and the basic replication and reproduction of effects that we always do with EEG research are these sensory event-related potentials. So essentially, if you sensory or other event-related potentials. So essentially, if you have a new EEG setup, you do your first EEG experiment, and you are not able to obtain visually evoked potentials from your data acquisition and data analysis pipeline, then something is really not working in your data acquisition and your data analysis, because these are very robust effects that have been reproduced over 60 years of research, and I have reproduced them myself, and I'm I don't know, I don't consider myself the greatest experimenter ever. So if I can do it, everyone can do it. So these things really are good as quality checks for your data. Everything else that then goes into, for example, explaining trial by trial variability in these potentials using computational models, or finding consciousness in one of the components as compared to not consciousness. So that's much more tricky. But the basic visually evoked potentials, motor sensory evoked potentials, and auditory evoked potentials, if you are not able to reproduce them in your own experiment, then something is not going quite right. So you always have a nice quality assurance if you reproduce them. So hence they are important.
Do you mean any visual stimuli, or do you mean a specific one?
No, essentially I mean any visual stimulus. So the typical thing, as we will see in a second, are checkerboards, which drive the system quite a lot. But essentially any visual stimulus that where you have first the screen is kind of, there's nothing happening on the screen, and then something is flashing up. So something like that, I mean. And the same for the other ones, but we will talk in detail about the stimuli. So these are sensory event-related potentials, which are really, really robust. I mean, they are clinically, they're used in clinics to monitor, for example, anesthesia depth. And if they wouldn't be that robust, of course, that wouldn't be done. Can you actually see that? Because you can lower the thing there, but that you guys need to decide that, whether you want to sit in a bunker. Quite light, but so that's the sensory event-related potentials, and then there's a class of event-related potentials that is usually referred more to as the other ones. This one. That's that, and then the other one.
There is something with your level.
There's another class of event-related potentials that's also fairly robust, but is not directly related to external stimulation, but related more to cognitive processes, although very basic cognitive processes. And these are the so-called cognitive event-related potentials, by which people usually mean mismatch negativities, P300, language-related event-related potentials, and error-related negativities. It's a bit better, but there's still, it's the door, right? The door makes quite a thing. But anyway. But we will be here for a long time, so the sun will eventually go away. And finally, there's a class of potentials that is movement related, and we will also talk about that. So this is really kind of the basic experimental toolkit and experimental results that are in EEG, that will always be there and that are really robust, and that mainly exist because the neural system and the brain is wired in specific ways. And basically these cognitive event-related potentials, they all work because the brain is mainly responding if something changes, and if things remain the same, it doesn't respond, but we will see that. And so the idea is to go about all of them and learn about them, starting with visually evoked potentials. So visually evoked potentials, the typical clinical setup is to use what are referred to as checkerboards. These are actually chessboards, but they are called checkerboards. Like local field potentials are just potentials, but are called field potentials. Somebody messed up always in the history of things, and then these kind of terms get stuck. So this is an example that I recorded during my PhD work. So we had a checkerboard on the left side of the visual hemifield which you cannot see here, but you have to imagine. So there was a checkerboard coming up at time 0, and then at 500 milliseconds it reversed. So everything that was white became black, and everything that was black became white. And actually these data were recorded together with fMRI data, so that was a simultaneous experiment. But nevertheless, we were able to record quite stable visual evoked potentials. This shows kind of the basic response for electrodes that are located on the opposite of the stimulation in the back of the scalp. So these are occipital, parietal occipital electrodes. I think here's actually the average of O2 and PO8. And what you can see quite strongly is the P100, so a positive on these electrodes, positive response at 100 milliseconds, and a negativity around 140 milliseconds. This is the response. So here's 0, the response to the onset. So there was a gray background, and then on the left side, in the left visual hemifield, this checkerboard came on. And then after 500 milliseconds, it reversed. So it didn't go from a gray background to a checkerboard, but from a checkerboard of where things are black and white to a checkerboard where things are white and black, essentially. And then you also get a P100 for this reversal. It's not as strong as to the onset of the checkerboard, but it's still there, and 140 is also still there. What you also see here is 2 contrast levels. So that's the C. Michelson. That's the Michelson contrast. It's just a way to quantify contrast, and black and white in this quantification has a contrast of 1. We also had a lower contrast condition, so where it was not black and white, but some darker shade of gray and a lighter shade of gray, and making up the checkerboard. And then the response to the checkerboard onset, and also to the reversal, you hardly see anything, but to the onset is weaker, showing that there is a response of the visual system that you can record at the scalp, but this response depends on stimulus properties, basically, like you already asked about. So in this case, what this shows is that for these kind of checkerboard stimuli, contrast definitely determines the strength of the response, and large contrast to something that is just visually very so it's fairly simple to predict what kind of things give you strong responses in the brain for sensory stuff, things that you feel like perceptually as salient compared to things that you feel perceptually not that stunning or salient, give you stronger responses. One can, so this actually here shows where, maybe I'll zoom in a little bit, the electrodes that we are talking about. So I meant electrode O2 here, which sits basically on the right to the central fissure on the scalp. One can study the dependence of these visual evoked potentials on other visual features. So what is shown here from the Niedermeyer book is the response in different electrodes with regards to spatial frequency. So of course you can have checkerboards that where you have very big tiles, so you have a low spatial frequency, so over the same amount of space the stimulus varies only very little, or you can have very high spatial frequency where things where you have very small tiles and things change a lot over the same kind of space. And what this is supposed to show a little bit is that there's kind of an optimal spatial frequency in the sense that a certain spatial frequency, so around this 31 checks, doesn't really matter what the unit is, but this kind of spatial frequency elicits, for example, if you look at this electrode, the largest response. And if you go to lower spatial frequency, it goes lower. And if you go, if you go to, is that higher? I guess that's higher. Higher spatial frequency, it gets not that strong. And if you go to lower spatial frequency, it doesn't get strong anymore. Basically showing that also the amplitude here of the P100 can be modulated not only by contrast, but also by spatial frequency. Now, these are kind of typical electrode views of data, which are useful to first get acquainted with this thing, but are also, from my perspective, a little bit misleading, especially if you think about that you record usually with electrodes all over the scalp. As we saw, you have these caps, and you have usually the normal system these days 64 electrodes. So what's happening with all the rest? And so these are different views that one can take on data, on EEG data. And this I call the electrode view. So this is actually from the SPM software visualization, but you will have similar visualizations whatever you use, or usually, or I mean, you can also simply just plot all of them in Python or whatever. Python, actually, there's also a nice EEG, or a good, let's say, good EEG suite called MNE. So here, basically, you have all electrodes stuck on top of each other. This I can never count. How many is this? Is this 32 or 64? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. It's more 64. And it's the same data as previously shown. So you have at time 0 the onset. And then you have the P100 around 100 milliseconds, and you have this reversal response after 550 milliseconds. And what I want to basically stress here already is that here in the parietal occipital electrodes you see this positivity, but at the same time, for example, in these frontal electrodes you see a negativity, meaning that it's called P100 because in certain electrodes that are of main interest for visual responses, their activity is actually positive, but in other electrodes at the same time you actually observe a negativity. Hence, this is why these P and N terms are okay if you have very few electrodes and you only record from them, but as soon as you have more electrodes, you see that at the same time, usually because of dipolar activations, you also have negativities. So the same thing is shown so this basically, if you just stack all the electrodes, the spatial configuration gets a little bit lost. I mean, you still have here parietal electrodes, so back of the brain, parietal occipital electrodes, and here your frontal electrodes. So there's still some spatial configuration here, but of course that's the more natural thing is to actually have them spatially arranged. So here this would be the front of the brain, this is the back of the brain. As you can see here, if you can see this, if you focus on kind of this these sets of electrodes, the stimulus was presented in the left visual hemifield, and so the strongest responses you see around here. So look here, for example, and here, and then you also usually have a lot of electrodes where not much is happening actually. So for example, here these electrodes and the central electrodes, there's not much happening. Yeah. So these are two other ways to look at these data. This, from my perspective, the simplest way to look at EEG data is what I call the data matrix view, because that's essentially how you would also store these data in working memory of your computer, and then if you want to explain all of the electrode data in peristimulus time, this would be essentially the data that you model. Again, this is very similar in a way to this what you here have on the left, with the difference that instead of having individual lines, you have color codes for peaks and valleys. And again, this is the same data. Here you have all the electrodes, a little bit smaller maybe. So starting here with frontal electrodes and central electrodes, temporal electrodes, and so on. And again, here you have the 100 milliseconds post-stimulus onset, and you see that there is there are some positivities. So here, so what is usually referred to as P100. Not in O2, but there's also a lot of negativity at this time. And yeah, so from a programming perspective, this is the most natural way to represent EEG data, even if it's maybe not the most intuitive from looking at it. I think the most intuitive from looking at EEG data is actually what follows here. So this is what is usually referred to as a topo plot or topography plot. It's in a way similar to this kind of approach, with the difference that instead of showing the entire trace for each electrode, this focuses on a single point in time. Yeah? So this shows for all electrodes the potential at 0 milliseconds, where you see there's not much going on, especially if you compare to what happens if you look at 100 milliseconds. So you see here on the left there's this line, and this just jumped from 0, where everything is kind of greenish, to the P100 peak. And then you can see quite clearly what I meant by this dipolar pattern, and the fact that you have positivity over certain electrodes, so red being positive, and negativity over other electrodes. This, of course, you can quite easily imagine to be due to a dipole that projects like here, although this looks more like, I don't know where this is, but that you have one dipole at a certain area in the brain that has a negative and a positive pole, and what you observe on the scalp is the positivity and the negativity of these two poles. So let's go back there. And this, of course, you can follow up. So if you go to 140 milliseconds, you see this negativity mainly on the contralateral side. So again, here also to make this transparent, this is I don't know whether that's that clear, but this is the front, this is the back, this is left, and this is right. And the stimulus was presented in the left visual hemifield. So you have the strong response in the right occipital cortex, like Rainer Göbel also mentioned. Yeah, this relates to the functional anatomy behind that. So I guess you all are familiar with the visual system. I guess who's not familiar with, who wouldn't know about the optic chiasm? Anyone who has not learned about the visual system? Chiasm? No, so you're all familiar with this. Basically, the notion that the left visual hemifield is projected to the right primary visual cortex, and the other way around, due to the fact that certain axons from the retina cross, and certain, so the ones in the medial retina they cross, and the ones on the lateral retina they don't cross. So this is, of course, why you observe this strong lateralization of P100 effects. This is also a nice visualization of the visual system. And the only thing that I want to highlight here is that there is more than primary visual cortex, which we will also have a look at later. So primary visual cortex being really at the calcarine sulcus and being mainly to the medial side, but then you have these secondary areas and things like the lateral occipital complex, which is involved in object recognition, and things like MT or area V5, which we also saw on Wednesday as involved in motion detection. So I'm just bringing that up because we will see LOC or lateral occipital complex in a second. This is how it looks from the medial surface. Again, this is where Rainer Goebel now at Monkeys put these stimulating electrodes. Good. So after bringing this up, now one other thing. So what we presented here, and which is a typical thing, is that we presented full hemifield checkerboard, and then the first response that you really strongly observe is the P100. You can, however, also observe an earlier response, which is the so-called N70 or C1. However, for this you need a slightly different experimental setup. Namely, you need to stimulate only 1/4 of the visual hemifield. So the basic idea is as follows. If you are in this case, the right visual hemifield, of course, what you present here projects to the left primary visual cortex, which is shown here. Looks a little bit weird, but just think that this is calcarine fissure and showing the left visual cortex. And now the additional thing here is that if you have something that is presented in the upper visual hemifield, this is projected to the lower bank of the calcarine fissure, and the other way around for something that is presented in the lower half of the visual field, that's projected to the upper upper bank of the calcarine fissure. So again, things cross over, like left and right visual hemifield being crossed, also upper and lower visual field being crossed. And we, of course, because we had full hemifield stimulation, we did not have this stimulation of only the upper field or the lower field. If you think that these kind of activities induce a dipole in the cortex, that makes up the visual primary visual area, then and you think that the pyramidal cells in there are all aligned, of course, with that dendritic tree towards the cortical surface and the axon towards the white matter, then these dipoles that you get from the stimulation of the upper and lower visual quadrants, they, because of this anatomical arrangement that the primary visual cortex is folded around the calcarine fissure, leads to dipoles that are perpendicular, not perpendicular, that are oriented in opposite directions. So one of them is here oriented upward, the other one is oriented downward. Now if you, then it of course still depends where the plus and the negative is, but let's say the y gets a positivity here, so then let's say that the arrowhead is the positivity. So if you record a stimulus in the, so if you present a stimulus in the lower right quadrant, the idea is that you can observe a positivity at this electrode. If you present a stimulus in the upper right visual hemifield, you observe a negativity, because just the dipoles, although they are within cortex, of course they have the same orientation, but because of the cortex being folded or wrapped around the calcarine fissure, they get opposite orientations, and then you would get additional negativity here. And then the story is that when we presented our full hemifield thing, these two, of course, occur at the same time and cancel each other out. So the thing about the N70 and C1, or the N70 aka C1 amplitude deflection of the visual evoked potential is that you need upper or lower field specific stimulation, and it is referring to the earliest response of the visual system, so the primary visual cortex, which also means that this big peak that we saw, like the P100, is not the response of the primary visual cortex, actually. But it's rather due to, although this is, yeah, I think one can deal with it. It's rather due to things that are more lateral. So the way to read this graph, so this is from actually a nice review on P1 and N1. No, it's actually it's a research paper, it's not a review, that tried to look using dipole modeling into the question of where do these things that we see on electrode level actually come from. And so they used dipole models. We'll talk about later what dipole models really are. But for the moment, just think that, yeah, basically you put hypothetical dipoles somewhere in the brain, and then you ask how active are they given some observed scalp series. And the point here is that the late P1, around this time, and also early P1 activity is explained much better by more lateral dipoles than by central dipoles. Again, meaning that this C1 activity, which they could here look at because they had lower left quadrant stimulation, relates to primary visual cortex, and the P1 activity relates to activity in higher extrastriate areas, like it's then always said, essentially things like lateral occipital complex, even if there's not a real object visible. Maybe one last thing about visual evoked potentials, I guess, that they can not only be manipulated by the properties of the stimulus, like contrast or the spatial frequency, but also things like arousal or attention. So here an example from arousal. So here the story is that people were asked to respond as quickly as possible. Then they would have medium arousal, and if they then are told to respond even faster, then the P100 amplitude gets even stronger. This is similar to basically increasing attention. So this is also some work from my PhD thesis, where we had people spatially attend to stimuli or not. Details don't matter right now, but what this shows is that these full lines, of the P100, the amplitude is larger if people attend spatially to the upcoming stimulus than if they don't attend. So, meaning that the amplitude of these quite early sensory components, not the very first one, which is C1, but kind of the second visual response, can be modulated by internal factors like attention. And if you remember what Jakob told us on Wednesday, there are certain people like Carl Friston that relate attention to the precision of top-down predictions, whatever. So it's not really clear what attention is. There are some theories about it. Experimentally, if people attend to it, they are faster, and the visual responses are stronger. I think I skipped it. So one last thing about visually evoked potentials. I'm almost like Rainer Göbel. And this will be my last example. No, now I have one more example. There is something called the N70 or vertex positive potential that relates again to the stimulus, namely the type of stimulus, so faces versus cars. So faces are, of course, very important stimuli for humans because we do our social interactions mainly via faces. So you find in the brain, as we also saw on Wednesday, in certain areas you find stronger responses to faces than to other objects. And this is very famous in fMRI. So there's something called the fusiform face area, which responds more to faces than, let's say, cars or or locations. There is something similar in EEG, namely the N70 or vertex positive potential, which is the same thing. So it's just again that it's has its negativity at the lateral side, and at the same time there's a positivity at the vertex, which is just the highest point of the skull. So basically saying that these face selective responses in fMRI can also be observed in EEG and are there referred to as N70 or vertex positive potential. So summary, and these are kind of the summaries that you should kind of learn by heart, so that you now always know if somebody says, and then we looked, for example, we correlated P100 activity with something, then you know what they roughly mean by P100. So C1 and 70 strongest in midline electrodes and likely generated in primal visual cortex. Polarity varying as a function of upper and lower visual field stimulation, and onset latency around 40 to 60 milliseconds, peak latency 80 to 100 milliseconds per stimulus, something around 70 milliseconds obviously. P100 largest in lateral occipital electrodes, generation mainly dorsal extrastriate, maybe even ventral fusiform gyrus, and also strong, or maybe a little bit more strongly modulated by cognitive factors than C1. That's something that you might find more often, that very early sensory responses are harder to modulate by cognitive factors, and later ones are easier to modulate by cognitive factors. However, there will always also be a paper that shows that the very earliest responses are also modulated by certain cognitive factors. But as a pattern in terms of ERP literature or general neural responses, very early is not that easily modulated by cognitive factors. But like I said, there will always be the paper that shows the opposite. N140 also modulated by a number of factors and just peaking later. Another thing that you will find for all of these ERPs is that the generation and the source of the earlier components are usually much better understood than that of later components. So I think nobody really can say what N140, it's not like this is V3 or V2. This is more like the summed response of more visual areas involving definitely not only the primal visual cortex, whereas early amplitude deflections in ERPs are usually easier to pinpoint. And mainly that is because they can be explained by, let's say, only a single dipole, and then everybody believes, okay, then this pattern looks like a single dipole, then I guess the pattern is from this area. And finally, in 70, a phase selective potential deflection. It will make sense here. Good. So that was basically like the first potential that we discussed in some depth. Now you will find that the remaining ones, the presentation and also the knowledge that comes with them follows similar lines. Good. Any questions right now? No. Good. Then let's have a break and